All right, let's continue where we left off last time. Last time we were talking about development of neuronal identity. We were discussing also when a cell becomes post-mitotic in the neuronal system. Right? If it's post-mitotic, that means now the cell has basically been committed to a specific function and role, right? And then they become a specific type of cell, right? It could become like a oligodendrocyte or just a regular neuron or any of the other glial cells like an astrocyte. So that's what we mean when we say post-mitotic, and that's basically establishing the actual neuronal identity. It's also going to be determined by different types of things that come into play, right? The transcription factors, we talked about those already, right? SOX, OCT, all those other transcription factors. Ligands and receptors. So we're going to talk about ligands and the receptors. Ligand is like the whole lock and key thing that you guys probably remember from A&P, right? Ligand is the key, and it needs to bind to a certain receptor. And so that's going to be a very important aspect of neuronal identity as well. So we'll talk about some of those in a little bit. So here's just an image that basically just gives you an idea of how some of these different genes and transcription factors and things like that come into play. So over here we have sonic hedgehog. Right? Sonic hedgehog is an important gene that allows for the migration and distribution of cells. Right? For this image over here, they highlighted the sonic hedgehog signals, and you can see where you have the notochord as well as the floor plate, and that's in the developing embryo. And then also other things that come into play. Remember BMP, bone morphogenic protein, which is going to be antagonized by noggin. Right? Noggin is going to basically be one of the regulators of BMP. And so these are all going to be really important, again, for establishing migration patterns of cells as well as the actual neuronal identity. And so noggin helps to basically preserve the neural identity of the ectoderm, and it effectively inhibits the ability for BMP to function properly. This is a slide from your text that talks about all the different types of genes and ligands and transcription factors that come into play for things like expression of the genes, for repression of the genes, precursors, etc. So this is just kind of FYI. You don't need to commit any of these to memory. It's just kind of a depiction of how these interact together to establish neuronal identity. So now let's get into some of the things that might happen clinically speaking if you have any mutations or deficits in these types of genes. So this is a really, really sad condition. This is holoprosencephaly, which effectively is a mutation of the sonic hedgehog gene. And sonic hedgehog is going to be really important again for like cell migration, for movement of the cells as they develop in the embryonic phase. And if you have a mutation of the sonic hedgehog gene, that means cells aren't going to be able to move properly. And so holoprosencephaly basically prevents the ability for the cerebral cortex to turn into two separate hemispheres. And so basically you get one giant hemisphere of the cerebral cortex, and it also messes with a lot of the bones and the other structures in the face and head to be able to move properly. And so really sad condition. And there's other conditions that we're going to discuss. So spinal bifida. Spinal bifida is a neural tube defect. And there's different types of spinal bifida, which we're not going to go into the different types here, but there's a couple. There's spinal bifida where you're not really necessarily seeing the protrusion of the spinal cord out of the skin, but there's other ones that you can see protrusion of the spinal cord out of the skin, as in this case. So this is a more severe form of spinal bifida. There's spinal bifida occulta, which is whenever you hear the term occult, right, it's like something that's kind of hidden right beneath the surface. You don't really see it. But with spinal bifida occulta, instead of seeing this like red protrusion or I guess also, what would you call that? A herniation of the spinal cord. Instead of that, you only see the skin. You'll see like a hair tuft. It's like a bunch of hair that grows right there. Don't ask me why you see a hairy tuft. It happens for whatever reason. I'm not sure why. but that would be spinal bifida occulta. But anyways, what's the underlying cause of spinal bifida? It's a folic acid deficiency. So folic acid, when do you take folic acid? If you're a lady, especially. Yeah, it's part of your prenatal vitamins, right? If you're planning on having a child, it's really important to have enough folic acid so that the child doesn't have any neural tube defects when they're born. As an adult, folic acid is also really important as well. You don't want to have a folic acid deficiency because it can result in anemia, right? So folic acid deficiency looks a lot like vitamin B12 deficiency. They both cause anemia. But in the developing womb, with the developing fetus in the womb, you need to have folic acid in order to actually develop the spinal cord properly. Anencephaly, that's an individual that's born basically with no brain. Holoprosencephaly, again, we talked about that as a mutation of your sonic hedgehog gene, and so you're not going to get migration of those neuronal cells. Aniridia is where you have destruction of the iris or malformation of the iris. And then there's also aneuploidy disorders like Down syndrome, which can result in multiple different types of phenotypic expression, right? With Down syndrome, it's a syndrome, right? Whenever we talk about a syndrome, it's a disease that affects lots of different parts of the body, and you have a constellation of symptoms. So with Down syndrome, shorter stature, there's going to be varying degrees of cognitive disability with Down syndrome, atrial septal defects, ventricular septal defects in the heart. They also have predisposition for Alzheimer's, predisposition for certain leukemias. So whenever you hear about a syndrome, that means you have tons of different symptoms being involved in that. And so aneuploidy disorders that affect your karyotypes, this is trisomy 21, that's going to be Down syndrome. So here's another image of, actually that's the same picture I showed you guys earlier of holoprosencephaly, but then you can have varying degrees of severity of holoprosencephaly. But the main thing that you see with holoprosencephaly is that with a normal brain, you get your hemispheres, right and left hemisphere. With holoprosencephaly, you get varying degrees of a lack of separation, right? The most severe one is going to be the alobar, so you have no lobes to the brain. So pretty nasty condition. Yes?
So the condition aniridia with the iris, does it affect someone's ability to constrict or dilate their pupils?
Oh, yeah. Yeah, if you don't have an iris, you're not going to be able to constrict and dilate your pupils. Yeah, so your eyes basically stay permanently dilated. Yeah, so that would cause, over time, that could potentially cause damage to the retinas as well, right? Because one of the reasons why your eyes constrict and dilate, one of the reasons, is when you're outside and you got plenty of light, right? You don't need your eyes to dilate. If you go to the eye doctor and they dilate your pupils, right? You guys ever had that done before for an eye exam? So they put like little, do you know what they actually use for dilation of the pupils? They use cocaine. So cocaine is actually a Schedule 2. It's either Schedule 2 or Schedule 3 drug. It has certain medical applications. One of the applications is in optometry. They go ahead and dilate your pupils using liquid cocaine. It's like a very low dose of it. But one of the things that cocaine does, it's an alpha 1 agonist, so it's going to cause dilation of your pupils, right? So when they dilate your pupils, after you get an eye exam, what do they give you when you get an eye exam like that? Have you ever done this before?
No.
They'll give you these like grandpa glasses, right? The big sunglasses that just wrap around. They look so completely hideous, right? But it's to protect your eyes because you can't get any UV light. Otherwise, it's going to cause damage to the retina. And so, yeah, with a condition like, huh? Really? Just with the lights? Okay, yeah. Well, I had one eye exam where I had to wear the grandpa glasses. I can't remember what it was. It was years and years ago when I was younger. I got bad eyesight too, so maybe they were like trying to see if there was any issue with my retinas. You had a question?
I don't know if this is a dumb question, but that's their eyes, right?
Which one? Yeah. Oh, yeah. With the holoprosencephaly, that's the eyes. Yeah. So you're not only getting poor migration of the actual cerebral cortex, the cells in the cerebral cortex, but remember, a lot of your ectoderm and the neuroectoderm makes up the bones of your head and neck, or part of your face. right? Your frontal bone, the maxillary bone, all those different bones are derivatives of the ectoderm. And so, yeah, if you're not getting proper migration of the cells in the ectoderm, that's basically, you know, it's like a cyclops, right? The eyes basically just kind of stay in one spot. So really horrible, horrible condition. I don't know what the survival rate is of that. Let me actually look that up real quick. Yeah.
How do you go about that? Even if your brain, for example,
Oh, my internet doesn't work. Can't even look that up. Actually, the internet on the computer probably works. Let me look that up real quick.
like if someone's born with no brain, what is that?
The child doesn't usually survive with anencephaly. There's been cases where they can survive for like a little bit, but so with Holland prosencephaly, 20% survive beyond the first year of life. And of those 20%, I imagine that not many of those actually survive. Now, with a lot of genetic disorders like this, usually the child dies in utero, right? They'll usually be miscarried. A lot of miscarriage are due to the fact that there's actually an underlying genetic disorder where the child wouldn't be viable outside of the womb. So what was the other one that you guys were asking about survival rate? So we looked at Holland prosencephaly and encephaly. Encephaly, survival rate, because I don't have these numbers off the top of my head. So 75% of infants are stillborn. 25% will die within a few hours, days, or weeks.
So 0.
Yeah, basically 0. There's a video that I saw of...
That baby looked a little old, though. Unless that's a fake picture.
It's not a fake picture. There's been a couple cases where the kids can live a little bit longer. There was like this one case I saw, I think it was in like India or something, where a kid was born with anencephaly, and they were being kind of like paraded around the town because they're like, no one's ever seen anything like that before over there, right? So, yeah. Usually the kids don't survive, but I guess there's a couple exceptions, right? This kid definitely does look a little bit older. You're right about that. Any other questions about that? Yeah. So Holland prosencephaly, long story short, it's a sonic hedgehog mutation. And retinoic acid, albeit is important as a transcription factor, you know, for a lot of different things, it can be a teratogen under certain circumstances. So retinoic acid has been known to basically affect the sonic hedgehog gene, causing mutations. And if that happens with Holland prosencephaly, again, you basically have a defect in your prosencephalon. Right? Remember your prosencephalon, that's what produces eventually your telencephalon as well as your diencephalon, which eventually turns into your cerebral cortex, thalamus, and hypothalamus. And so these areas are, these genes are going to be really important to help determine the migration pattern of those regions in the brain. And again, also affects the bones in the face as well. So here's a couple images that depict normal versus abnormal development of the brain in fetus. Right, on the left side, you're going to have normal development. And so in those cases, retinoic acid has been, beneficial, right? It's been beneficial to allow for the migration of different subsets of cells as well as genetic expression. However, if mom takes retinoic acid during pregnancy and you get an excess of retinoic acid, then that could basically affect the development of the brain and other regions of the head as well. And here's another image again on this little slide over here. B, you got normal brain on the left, and then you have the abnormal Holland prosencephaly brain on the right. And then here's human brain. Human brain on the left, that's normal. So that's regular, normal, late gestation brain compared to Holland prosencephaly brain in a human. So again, these are going to happen if mom takes retinoic acid in excess, right? So that's one of the deficiencies there. Yes?
So if we take normal levels of it, it'll be fine.
I don't know what the cutoff is of retinoic acid. Like, what's the safe? How much retinoic acid is safe?
Where is retinoic acid?
It's vitamin A.
Isn't it an Accutane?
Yes, it is part of Accutane. Yeah, so retinoic acid affects basically the skin and regrowth of skin. So when you take Accutane, it allows for your skin to turn over a little bit faster. And so it can help to address things like, really like bad cystic acne and things like that. And yeah, so retinoic acid is part of Accutane. What's this say? Okay, so total vitamin A intake for a pregnant woman is over 1,500 IUs per day. IUs are international units. Yeah, that'd be 4,500 micrograms. Yeah, I guess that's the safe amount. I don't know. Don't quote me on that. I'm not going to test you on what's the safe amount of retinoic acid to take to prevent prosencephaly in your kid. So, yeah. Anyways, in terms of cancers, medulloblastoma, this is a cancer that affects the cerebellum, and it's going to be the most common brain tumor in children. And what's this involving? It's also involving the sonic hedgehog gene, but it's going to be specifically involving GLI1. And GLI1 is going to be what basically is regulated by the sonic hedgehog gene. But if you have too much GLI1 expression, then that's going to cause medulloblastoma, causing tumor within the cerebellum. This image over here, also from your textbook, shows how the cerebellum cells are going to migrate during histogenesis. And you can see the cell types that are going to be within the granule layers that are going to eventually develop and grow into the more matured cells. And that's going to be dictated by the sonic hedgehog gene, right? So it allows for the cells to migrate properly. And then you have your cells that are undergoing mitosis, and then eventually those cells would become post-mitotic. But if you have a GLI mutation, then those cells are going to continue to develop and grow, right? So you get too much mitosis of these types of cells, resulting in medulloblastoma. So with cancers, basically what's going on with cancers is that cancers circumvent the cell cycle, right? You have different stops within the cell cycle that prevent the cell from continuing to undergo DNA replication and synthesis, and then continue on to undergo complete mitosis, right? So you have these basically stops in place so that you can help to regulate the cell cycle. Whereas with cancers, they circumvent all that. Now, in terms of normal neurogenesis within the cerebellum, you're going to have the pile surface or the pile surface. That's going to be where the pia mater is. That's going to be on the outside of the brain. And then you have your neural tubes. This is like during neural development. And so you have the neural tube lumen, so the opening of the neural tube. And cells are going to be migrating from the neural tube lumen up to the pile surface, then back again down to the neural tube lumen. And so that's going to be taking place at different stages of the cell cycle. So in this next slide here, it talks about those different stages. So you have your G1 stage, and you're going to have cells mostly hanging out near the lumen. We call that also the ventricular zone. And then as the cells undergo DNA synthesis, that's what the S phase allows for, or what the S phase represents. DNA synthesis, where everything is basically being doubled up, right, in terms of its genetic makeup. And then it's going to be hanging out around the pile surface. And then as it goes back down to the G2 phase, it's going to move down into the ventricular surface again near the lumen. And then you have the completion of mitosis. And so that's all taking place within the developing brain, right? Cerebellum, as well as parts of the cerebral cortex too. Again, when you have termination of the cell cycle and the cell becomes post-mitotic, that's it, right? The cell is now going to have its final, has its final destiny, basically. It's going to be one of the types of neuroglia, or it's going to be an actual neuron at that point. What are some of the things that allows for the cells to undergo differentiation and replication? It's going to be delta notch signaling. Delta is going to act as the ligand, whereas notch is going to be the receptor. And so when those come into contact together, that's going to allow for differentiation. And it's going to allow for expression of the Hox genes. Right? Remember those Hox genes, the homeobox genes, allows for like segmentation, differentiation, all that. So this is going to be one of ways that the cells communicate with each other is through this type of signaling. So that's delta notch signaling. And then after delta notch signaling takes place, then you have a downstream cascade of effects, including transcription factor regulation, synthesis of different proteins that allow for the cells to actually become fully differentiated. So generation of neurodiversity and differentiation of different cells. So you have cell-to-cell interaction. One cell might have, for example, ligands on them or release ligands, and then the other cells might be receiving those signals. And then so you have those types of interactions between the cells. You also have diffusible signals. Diffusible signals. We're going to talk about these types of signals when we get into the developing of like neural circuits in the next portion of the section. We're going to talk about diffusible signals, non-diffusible signals. And so noggin, chordin, remember we talked about those already. Those are the ones that are going to be regulating bone morphogenic protein. And then you also have transcriptional codes coming into play, which basically allows there to be like a history of different types of processes taking place within the cell. And then you have the completion of all the division, and then that's going to be where you have the post-mitotic cell. So here is an example of how the cells will basically be turned on, allowed to be differentiated, and to migrate into their respective locations. So you have your cells in your ectoderm, right? Part of your neuroectoderm, all those the major cells that are going to turn into the spine and brain. Sorry, the spinal cord and brain. And then you have your different types of cell signals coming in. So bone morphogenic protein, noggin and chordin, which can also affect bone morphogenic protein. Then you have things like Sonic the Hedgehog or Sonic Hedgehog, SHH, coming into play to allow for movement of the cells, differentiation of the cells. And then you also have orientation of the cells too. So there's going to be cells that are going to be in the dorsal section. There's going to be cells in the ventral section. And then you finally have the ability of full differentiation of the cells, and that's going to be the process of neurogenesis. And then you have oligodendrocyte formation, you have astrocyte formation, and neuron formation in these processes, right? And you have all sorts of different transcription factors coming into play to affect neuronal differentiation. So this is the specification of your oligodendrocytes, astrocytes, and neurons. That's going to be the final step in this whole process, right? Now those cells are going to be post-mitotic. That's their final destination. Here's another example of what also can happen in terms of migration of cells. So cells can segregate into different layers. So we're going to learn about these layers later on when we go into more detail about the cerebral cortex, right? You have your internal as well as external pyramidal layers, right? You have the pyramidals, they're kind of like shaped like little pyramids in the cerebral cortex, right? So there's 6 different layers in the cerebral cortex. The retina is also going to have multiple different layers too. So we're going to learn about vision later when we discuss special senses in this class. So we're going to learn all those different layers and how those function. You got your rods and cones, and then you also have your the photo pigment receptors or things like that. So we're going to talk about all that later on in special senses. But when you have migration of these layers, that's going to be determined again by transcription factors, and it can also be determined by timing as well. So some cells might migrate at different times, some cells might get turned on at different times, and that basically determines what layers those different cell types are going to be when they are fully matured. Right? So don't commit any of this to memory. This is just an example of how important the transcription factors are when it comes to determining the destiny of these cells. All right, epithelial mesenchymal transitions. We're going to talk about how you have your epithelial derivative cells, right? Those are going to be the precursors, and then those can eventually turn into mesenchymal cells. And mesenchymal cells can also turn into a bunch of other cell types as well. So they're kind of, they're like a multipotent type of stem cell, effectively. So a step below pluripotent cells, right? So if you had pluripotent cells, those can basically turn into any tissue type. If you have multipotent cells, they can turn into different cell types within those three major layers, right? The ectoderm, mesoderm, endoderm. But anyway, so epithelial mesenchymal transitions, or EMTs, they effectively enhance the migratory capacity. Invasiveness, they are also going to be involved in apoptosis and also the production of extracellular matrix components. So in other words, these types of cells are really important for migration patterns, signaling between cells, and secreting things like signals, right, diffusible signals, and modification of cytoskeleton. Now, there's another cell type, and I didn't put this one in your guys' lecture slides, but the book makes mention of mesenchymal to epithelial transitions. So those are a little bit different. That's going to be going in the reverse direction. So you're having a cell going from a like multipolar spindle-shaped cell back into an epithelial cell. So it's basically rewinding the clock. But we're not really going to talk too much about that process. We're going to more talk about epithelial to mesenchymal transition. So here's your epithelial cell. And then you have different signals that come into play to turn that cell on and to allow it to effectively differentiate into a mesenchymal cell type. And like I said, mesenchymal cells are going to be really important to turn into, for example, like other cells, as well as like establishing migration of cell types to different layers. All right, now these are some of the things that might come into play when we discuss diffusion factors or diffusible factors. So diffusible factors can be anything that's basically allowing for cells to communicate with each other, right? Hormones, those are going to allow for cell-to-cell communication. Hormones take a while, right? Hormones are the whole entire endocrine system is kind of similar to the nervous system. As a matter of fact, there's a lot of crossover with endocrine glands, right? Think about the hypothalamus, that's an endocrine gland, the pineal gland, right? These are all found within the brain, right? So, but as opposed to normal neuronal communication, which happens really rapidly and very fast, hormonal communication is going to take a little bit longer, right? So it's going to be more long-term communication. But really important to allow for cells to do their thing, get turned on, and then eventually differentiate into different cell types. There's also morphogenic factors, and then there's also growth factors that come into play in these processes as well. And here are some of the different cell types. So these are all derivatives of the neuronal crest cells. And so you have sensory neurons, autonomic, parasympathetic neurons, melanocytes. You also have your adrenals, right? The chromaffin cells that you find within the adrenal medulla, those are going to be derived from your neural crest cells. Now remember, neural crest cells, those are the ones that eventually like migrate down above, so they start kind of like above the neural tube, and then they migrate their way down towards the rest of the body. And those eventually turn into the peripheral nervous system, right? Peripheral nervous system, autonomic nervous system, sensory neurons and whatnot. And so these are all the different cell types involved in that. So notice you have your neural crest progenitor, you have your bone morphogenic proteins, you got the WNTS transcription factors coming into play, and then you have other things that come into play as well. So glucocorticoids are going to be involved in allowing for chromaffin cells to mature, right? You have different stem cell factors that affect melanocyte formation. Fibroblast growth factor, FGF, that allows for certain cells to turn into adrenergic or cholinergic neurons. So these are all derived from the neural crest lineage. What else is important here? So migration in the central nervous system. So these cells are going to be eventually turning into, for example, GABAergic type cells. And these GABAergic cells are going to be found within the cerebral cortex, hippocampus, olfactory bulb. GABA, what does GABA do? Do you guys remember from AMP? If you don't, it's fine. We're going to be talking about it again when we discuss neurotransmitters. Yeah, it's basically an inhibitor. GABAergic cells are going to release GABA, which causes neurodepression, right? It reduces signaling, so it basically turns things off. It's a really, really critical neurotransmitter in the nervous system. And so these cell types are going to be involved in the cerebral cortex, hippocampus, olfactory bulb, etc. And again, once they become postmitotic, that's it. There's no more evolution in those cell lineages. Already said that. Let's move on. Cool. Let's talk about migration of cells. Right? We talked about the genes involved in there, but what about the physical things that allow for like scaffolding of cells as the cells move up and down? Right? There's actually like scaffolds that come into place. So within the cerebellum, you have scaffolds that are going to be produced by the Bergman glial cells. It basically allows for those cells to move up and down within the cerebellum, the different layers of the cerebellum. Same thing with the cerebral cortex. You have scaffolding that's going to be provided by radial glial cells. And radial glial cells have these really long processes that allow for cells to kind of like twirl around the scaffold so they can move up and down the different layers of the cerebral cortex. So I'll show you some images of what that looked like in a second. So this is within your cerebellum. So you have your granule cells. Those are going to be your, kind of your precursors, and they're going to undergo mitosis, right? They're still able to divide. They haven't yet reached their post-mitotic state. And as they're developing, they're going to be using the Bergman glial processes as scaffolds, so they can move up and down the layers of the cerebellum. And then again, you also have a similar process taking place within the cerebral cortex. You have your scaffolds produced by radial glials, right? They're going all the way up and down the entire length of the cerebral cortex, all the different layers. And then it allows for cells to basically migrate up and down, depending on which layer they're supposed to be at. Let's talk about a couple other diseases that are involved in basically the inability for cells to migrate properly, right? Lissencephaly is going to be one of those conditions, right? There's some animals that have lissencephalic brains, right? Those are your smooth brains. But humans can be born with lissencephaly as well, and it's going to be due to different mutations and diseases that come into play. Right? Gyrencephalic means that you have like different gyri and sulci, right? That's what a gyrencephalic brain means. So, in other words, we have a gyrencephalic brain, normally, unless we have a lissencephalic brain. But again, some animals just naturally have this. I incorrectly thought the sloth did. I'm wrong about that. I always thought the sloth had a smooth brain. Sloths actually have a gyrencephalic brain. They just kind of act like they have a lissencephalic brain. But koalas have a smooth brain. A bunch of other animals have smooth brains too. So certain rodents, bats, manatees, certain primates also have lissencephalic brains as well. But if they do, they're not going to have, a very sophisticated higher cortical function, right? If it's gyrencephalic, you're going to have more cortical function, or better higher cortical function. So in other words, mutations can cause these diseases like lissencephaly, and sometimes it's just a single gene mutation, right? You have a single gene mutation that can disrupt everything in terms of neuronal processing. So you can have mutations that affect the placement of cells, the timing of cell development. It could affect different types of proteins, of course, the ability for the cell to climb. Remember, we have your scaffolds. Bergman's glial cells, radial glial cells. If you can't provide scaffolding, those cells are not going to be able to migrate properly. And so 3 of the major genes that we're going to talk about are relin, LIS1, and DCX. Relin, when it's normally functioning, relin is supposed to help with migration of neurons. So neuronal migration, the branching of dendrites, as well as the dendritic spine formation, synapses, right? Synaptic formation. So in other words, if you have a mutation in relin, you can't do any of those things. By the way, that's a new addition that I added to the slides, so I didn't update your guys' slide, what relin actually does. But it's not a big deal. All you need to know is that if you have a relin mutation, that is going to affect the gyri and the sulci. So you're going to have basically poorly developed gyri and sulci. Now, worse than that, though, is LIS1 and DCX mutation. Any of these, if you mutate those, can cause lissencephaly. So LIS1 is really easy to remember, right, because lissencephaly. DCX, though, if you have a DCX mutation, that can also cause lissencephaly. And again, this is where you have a migration pattern. A migration error. You're not able to migrate the cells properly. And so basically the brain just remains smooth. It's malformed. That can cause cognitive impairment, mental retardation. That's a technical term, right? It's not a rude thing to say, unless you're saying that to somebody as a pejorative. But mental retardation is an actual, medical term. It's clinical. You can also get epilepsy from this too. So lissencephaly is not a good situation. So here's your migration of the neurons again. You got reelin, which is really important to allow for creation of your gyri and sulci. You have your glial cells, which are providing scaffolding for the migration of your neurons. So you have cells that are going to be involved in several different transcription factors, genetic expression. That's all involved in the ability for the cells to move and migrate. And so over here, the leading process is where you have the formation of your dendrites, right? The tailing process where you get the axon formation of the cell. And so if you have any issue with this process, then you're not going to be able to develop your brain properly. Now, see over here, if you have a LIS1 mutation or DCX mutation, that basically inhibits the ability for this to take place, right? So you don't have proper scaffolding and you don't have proper migration of cell types, causing the individual to be born with a smooth brain. And so here's a CT scan. I think it's a CT scan. Yeah, it's a CT scan. CT scan of a normal brain. So you can see you got your ventricles, a nice size. You got pretty nicely demarcated gyri and sulci. You got your gray matter. You got your white matter in the middle. But if you have any of those other gene mutations like reelin, LIS1, etc., then you're going to have disruptions of these processes. So this is what a reelin mutation looks like. Remember, that's really important for the defining what your sulci and gyri look like. So there are sulci and gyri in this. This is not a fully smooth brain, right? But the gyri and sulci don't look good, right? This is what they're supposed to look like. And if you compare a reelin mutation brain, you're going to see lots of disorders in the gyri and sulci. So disruption of your gyri and sulci. So over here, you got your blue arrow. That's going to be showing your ventricles. Over here, they're a little bit enlarged, right, compared to the ventricles in a normal brain. So you see some enlargement of the ventricles. Over here, you have these different tracts, right, and the white matter. And then your gray matter is up here, indicated by the red arrow. So that's your gyri and sulci, right? Now, this is what lissencephaly looks like. This is where you have either an LS1 mutation or a DCX mutation. It could be either or. Both of those will cause lissencephaly. And the radiograph, look at that. The brain is totally, completely smooth. So not a good situation, right? You have no differentiation between even gray matter and white matter. You can't even see the difference between the two. Right? The cells just have not properly migrated whatsoever. So the brain is completely smooth. Look at the size of those lateral ventricles. My gosh, those are enormous. Absolutely huge. So, terrible situation. Other diseases that affect the migration patterns of cells. Did I include this in your slides? You don't have Hirschsprung's disease on there? Dang it. I'll send that to you guys later today. Okay, sorry about that. I updated these the other day. I thought I sent you guys the updates, but my bad. Anyways, Hirschsprung's disease is another disorder that affects the GI tract. And so you're basically getting the inability to migrate the ganglia that are really important for innervation of the GI. And so if you're lacking the myenteric plexus, for example, then your colon is not going to be able to undergo contraction, right? You're not going to have movement of contents from your GI. Right? You're not going to get any peristalsis. And so Hirschsprung's disease is basically aganglionic megacolon. Why is it megacolon? It's because stuff builds up in there and it gets huge. You get this massive dilation of the colon. And you do see this more so with conditions like Down syndrome. And when a child is born with this, the parents are going to notice, oh, my kid hasn't passed the meconium. Meconium is the very first time a newborn baby goes number 2, right? So that's the meconium. If they don't pass a meconium within a couple days, then you're thinking, oh gosh, maybe this kid might have Hirschsprung's disease. It also could be caused by cystic fibrosis. That's not important for this class, but cystic fibrosis is a deficiency of a chloride channel gene, the CFTR gene. And if you can't produce chloride, or if you can't allow chloride ions to move properly, it affects your pancreas, it affects your lungs, it affects your skin, lots of different parts of the body. But cystic fibrosis can also be an underlying cause of Hirschsprung's disease. And then you can test this using an anorectal manometry, which basically tests the strength of the muscles within the rectum. And if there's basically no contraction whatsoever of those muscles, then you know you have a lack of ganglia within the colon. And there's different types of Hirschsprung's disease, but you're not responsible for the subtypes of Hirschsprung's. So I'm sorry, I forgot that I didn't send you guys the update. So I'll send you guys that update later today. So keep an eye out for that in the announcements. Any questions before we do quizzes? All right, let's get into it then. Are you guys having a hard time logging in because of the internet? Okay. Someone got in. That's a good sign.
Really annoying because I have Globe's Wi-Fi on my phone.
Really?
Yeah.
Are you sure it's not just connecting to your normal Wi-Fi?
I just checked. It says Lobos. That's so weird. And it's like working fast too.
Weird.
Things are choosing.
Yeah.
You have to be like in the correct area.
That's so lame. Cool. Everybody's in? Let's go ahead and begin. All right, awesome. Neuromeres and the homeobox gene. So great. Yeah, so the homeobox genes, also known as your Hox genes, right? Those allow for discrete, distinct, basically like, special units in different areas, right? So it allows for patterning of discrete areas. And so, yeah, those are going to be referred to as neuromeres. That's going to be the entire nervous system. So that was probably a softball question. Good. Yeah, so that's holoprosencephaly, right? Holoprosencephaly is going to be especially affecting some of the bones in the face, right? The cranial bones are all going to be affected. Right? That's why you get that kind of cyclops-looking presentation in holoprosencephaly. Anencephaly is not affecting the facial features. Anencephaly just means no brain. This should be a softball question too. Good. Yeah, those are organizers. Organizers basically affecting patterning, right? Patterning of the cells. It mostly affects the adjacent cells. So cell to cell communication. I take it you got that wrong. Are you gonna do that? Are you gonna do that during the test tomorrow? So yeah, that's let's see, which one is you? Is that you right here? Did you pick B?
I meant to do folic acid deficiency. Obviously, I should know that.
Yeah, so folic acid deficiency. That's what causes. What's this condition called? Close. That's the second part of it. Spinal bifida. Spinal bifida. Perfect. Yeah. So this is pretty severe spinal bifida. This is like the most severe type. Really nasty condition. It can cause all sorts of issues in terms of like neurological deficits on those those patients. They do surgery. It'll be like neurosurgery to basically put everything back inside where it belongs. So yeah, not good. So remember, retinoic acid is a teratogen. And now that you mentioned Accutane, that's one of the major things that if a person is going to start, well, if a woman specifically is going to start taking Accutane as a teenager, she absolutely cannot get pregnant because it is absolutely a teratogen. I totally forgot to mention that. Since you mentioned Accutane, that's one thing to be concerned about, right? And so if you do take Accutane and you are pregnant, that's what happens. You get mutation of the sonic hedgehog gene, causing holoprosencephaly. What's up?
What's a teratogen?
Oh, a teratogen is basically anything that damages the developing fetus in utero, right? So teratogens could be like alcohol, right? Lots of drugs, lots of medications can be teratogens, right? Chemicals as well can be teratogens. Hopefully that was a softball question. Medulloblastoma. Which part of the brain is affected by medulloblastoma?
Cerebellum.
Yep. The cerebellum. All right. Hormones, growth factors, extracellular matrix molecules. So not so much cytokines. Cytokines are ways in which cells do communicate with each other, but not in this regard. Cytokines, some good examples of cytokines would be like tumor necrosis factor alpha that induces apoptosis into a cell. IL-1, interleukin-1, interleukin-6 that allows for fever to take place. So those are good examples of cytokines. And neurotransmitters aren't really going to be coming into effect when it comes to neural migration. It's going to be more so growth factors and things like that. Looks like most of you guys got that correct. So this would not be a Reelin mutation, right? Reelin mutations wouldn't look like that. You would still have gyri and sulci, but they would be poorly formed, right? That's what a Reelin mutation would do. Reelin allows for like cell migration patterns and things like that to take place. And that's what would be. That's what would be the effect. You would still have gyri and sulci, just they wouldn't look great. But if you had a smooth brain, like what you see in this CT scan over here, that's what's going on with this kid. This kid has either it's a DCX or a LIS1 mutation, and that causes what condition? What's it called? Lissencephaly. Perfect. Nice. Yep, that's classic presentation of Hirschsprung disease. Newborn baby not able to pass meconium, right? And they're going to be eating, right? So they're still going to be producing fecal matter, right? So it's going to build up. It's going to cause toxic megacolon in those poor kids. I should just not. I shouldn't include photographs. I don't want to make it too easy for you guys. Yeah, anencephaly. So it's classic anencephaly. What's microcephaly? No, just small head in general. Small head. Yeah, they're gonna have a small brain. Pinhead. Is that her name? What's her name?
Are you talking about American Horror Story?
Yeah, she was featured in that. Right? There was a character that was basically her, but it was actually a normal. If you actually see the actress that played her, she's like really normal looking. She's actually scary. They did a really good job with the makeup, but the real character, like the real life, like actual circus performer, right, with a really tiny head. I think her name's Pinhead, if I remember correctly. Is it? Yeah, I think her name was Pinhead, but she was like a circus attraction, right? She was a very famous case of microcephaly. So.
They have like a normal brain function?
They're going to be slightly cognitively impaired, right? Yeah, they're not going to be like super high IQ or anything, but they're still able to function, right? You know, Pinhead was a successful circus performer. So cool. All right. Any questions before we move on to neuronal circuits? No questions? All right. Let's get on with it then. Let me just switch over. All right. So construction of neural circuits. We're going to talk about synaptogenesis, developing synapses, migration patterns of like the axon growth cone, right? That's how the axon, as the axon grows, it's called a growth cone. And then it's basically kind of like looking for dendrites to be able to come into contact with. So we're going to talk about all those different types of processes. We're going to also talk about the movement, for example, of neurotransmitters within the axon and how like dynein and kinesin comes into play with that. So we're going to discuss kinesins and dyneins in a little bit. Microtubule formation, as dynein and kinesins are going to be coming into play to move things either to the distal end of the axon or back towards the cell body. We're going to talk about lamellopodia and filopodia. Those are going to be portions of the axon growth cone that reach out and are able to basically make contact with dendrites. We're going to talk about some of the signals that come into play that either allow for attraction of the growth cone or repulsion of the growth cone, also adhesion to different surfaces. We're going to discuss tropic versus trophic. So tropic means basically like movement, whereas trophic means supplying the things necessary to allow the neuron to survive. So basically like nutrients, things like that, allow for the survivability of the neuron. We're going to talk about tiling, and tiling is kind of stacking of neurons and spacing out of neurons. We're going to talk about how synapses are formed. We're going to talk about the things that affect trophic interactions, right? There's different types of growth factors that come into play. So BDNF is a really famous one, brain-derived neurotrophic factor. And then we're going to talk about some of the receptors. Not super high yield. We're not going to go into all the different types of tyrosine kinase receptors, but we'll do a little brief overview of that. Again, we're going to discuss the Hox genes, and then we're going to talk about Sperry's chromo-affinity hypothesis later on. That's going to be towards the very end. So what's the very, very first step of allowing neurons to develop? And it's going to be neuronal polarization. So remember, neurons are going to be polarized. They're going to have, in general, cell body with their dendrites, and then they're going to have an axon coming off of the cell body, allowing for synapsing with dendrites or forming, example, the neural muscular junction. Right, so the neuromuscular junction where you have the neural end plate or the motor end plate to allow for the flexing of muscles. So polarization is going to be one of the most important steps. And again, these are going to be derived from your epithelial cell line. And so here's an image that's showing the polarization of neuronal cells. So you have in the red in the middle over here, those are going to be the apical aspect of the cell. Then you're going to have in the green area, this is the basal area of the cell. That's going to be your the bottom of the cell, basically. And those are going to be involving things like cadherins. So we're going to talk about cadherins later on. Cadherins basically allow for cells to come into contact and stay in contact with each other. So we'll talk about that later. Bless you. So the apical domain. Again, the apical domain, that's going to be on the very top of the cell. And over here in this diagram, you see that you've got little microvilli, right? Microvilli are going to be these little finger-like projections that come off the top of the cell. You're going to have tight junctions, right? Tight junctions basically don't let anything go through those cell layers, right? You have lots of tight junctions, for example, on your skin, right? In your epithelium, right? Tight junctions basically prevent water loss, water movement. Similar concept over here. Then you have Golgi apparatus, and Golgi apparatus is going to be really important for doing all sorts of things like repackaging different types of materials that need to be secreted by the cell. So they're going to be, repackaging different maybe neurotransmitters and things like that, and then putting them into vesicles so they can be released. So we'll talk about that later. And then you have your vesicular endocytosis. And vesicular endocytosis is how you'll have endocytosis bringing stuff into the cell and then turning those into vesicles so they can be transported throughout the cell for various types of functions. And then the basal domain, again, that's going to be at the very bottom of the cell. That's where you have contact with extracellular matrix. There could be blood vessels in there as well, so you can get nutrients and things like that. And then microtubules. Microtubules are going to be really important for internal cellular trafficking. Right? It's going to be able to allow for things to move around the cell. So super important. So you have endosomes. Whenever you have endosomes, those are going to be little tiny, basically like vesicles that can be moved around within the cell using microtubule scaffolding. Sorry, thank you. So again, neuronal polarization, first step of cell or circuit or neuronal circuit formation. And another thing that's going to be important in this process is a thing called partitioning defective proteins, or PARs. And those are basically scaffolding proteins and allow for polarity and polarization of the cell. So partitioning defective proteins are going to be important in this whole process of developing those cell connections.
You just said about what they're important for.
It's for basically allowing for polarization, right? Formation of axons, for example. And then these are also referred to as scaffolding proteins.
Okay, thank you.
So here's an image showing how you have development of the growing axon. So at the very tip, you're going to have an axon growth cone. And as the axon growth cone migrates until it can eventually find a dendrite to come in contact with to form a synapse, you're going to see it continuing to grow. And you have signals that are going to be coming into play that allow for amplification of this process. And polarity scaffolding proteins, again, those are going to be really important for this whole process. So they allow for polarity as well as the scaffolding of the cells. So here's neuroblast and neurites. Whenever you hear the term neurite, it's going to be any sort of projection coming off of the cell. So it could be axons, or it could also be dendrites as well. So that's what neurites are. There's going to be any type of projection. So as soon as a cell starts to differentiate, like, for example, the cells that were growing upstairs, after adding retinoic acid to the cell to allow it to grow and differentiate, you can actually see neurites coming off of the cells now. They're actually trying to form either synaptic connections or dendrites to allow for synapsing. And so as the cell begins to form neurites, eventually you're going to start seeing some significant axon growth as well. And so that's what's labeled over here in green. So they're able to use fluorescent staining so that it specifically targeted the axon versus the cell body or dendrites. And so again, polarity scaffolding proteins, right? These are going to be really important for establishing polarity and for establishing the ability to scaffold. So here's an example of normal functioning polarity scaffolding proteins, or PARs, versus if you delete or disrupt the ability for PARs to properly function, the cell's not going to form any neurites whatsoever. It's just going to remain as an immature cell, just a blob, basically. So you can disrupt PAR. You can do that through either overexpression and disruption through things like, or just basically disrupting PAR. That's when you get an ability to form neurites. Now, what's the whole purpose of an axon? An axon is basically to allow for connection to an adjacent cell. And what's the purpose of having those types of connections? So that you can allow for neurotransmitters to be secreted and released so they can bind to receptors on the other end in the postsynaptic cell. So what's the thing that allows, for example, neurotransmitters to be moved back and forth in the axon? It's going to be kinesins and dyneins. Now, kinesins, those are going to be anterograde. What does anterograde mean? It means going from the body to the terminal end of the axon. Retrograde is going to be going from the terminal end of the axon all the way back to the body again, and those are going to be carried out through dyneins. So kinesins go forward, dyneins go backwards. So let's watch a couple of videos on this. And they, by the way, the whole entire time, they're going to be moving along the microtubule. So this is an image where they actually were able to use fluorescent dyes to highlight the dynein kinesins moving forward and backwards. So let's watch another video. Let's watch that video once more because that was pretty cool. It's very short, but there's tons of videos like this that you can find online where you can see all the contents moving either forwards or backwards along the entire axon. So super cool.
So they can be considered like transporters.
Yeah, they're basically transport proteins. Exactly. And here's a really nice animation that shows what it looks like when you have dynein and kinesins. They literally walk along the microtubule, and they do this really fast, right? This animation is, way slower than what you would see in real life. And then these microtubules that you see that they're walking along, those microtubules are being broken down, they're being formed all the time, constantly, right? They're going in all sorts of different ways. I'm going to show you the dynamics of microtubules in a second. There's a really cool video that basically shows what it looks like when those microtubules are being formed, broken down, and redirected. So again, microtubules, that's going to be what provides scaffolding for dyneins and kinesins. And they're going to basically move all the way towards the tip of the axon, all the way towards the terminal end of the axon. You can think of that as being kind of a substrate. Right? The substrate is the thing on which or upon which a reaction is taking place. Right? So you can kind of think of this as being a reaction. Right? It's requiring, for example, ATP. It requires power, energy to be able to transport contents to the end of the axon back towards the cell body. Here's another video of kinesins.
Just like a busy city, there's constant motion inside of your cells. There's new construction, demolition, and most importantly, transporting goods from one place in the cell to another. Cells transport goods along cellular roadways. To transport cargo along these roads, the cells use motor proteins. Kinesin is one of these motor proteins. If you didn't have kinesin and other motor proteins, you simply wouldn't be alive. All the cells in your body depend upon these tiny motor proteins to organize and power themselves, to divide and multiply, and to communicate with other cells. Let's start with how you began, as a fertilized egg. That egg has to divide into many more cells. All of those divisions require kinesin and many other motor proteins. Development, forming tissues and different parts of your body, requires molecular motion and motor proteins. Every cell of your body requires them for survival. What we know now is that kinesin effectively has two legs, and these legs are able to coordinate a walking motion along a track. That track is called a microtubule, and the kinesin undergoes this beautiful choreographed walking action. While the kinesin is walking along this track, at the other end of the kinesin, it gets hooked up to a cargo. These motor proteins move quickly and efficiently. Relative to their size, they move as fast as a car on a freeway, but they're 4 times more efficient than your car in converting chemical energy into motion. We discovered kinesin in 1984. I was 25 years old at the time and a graduate student. I was interested in the transportation system inside of nerve cells. And what makes nerve cells so interesting is that they're extraordinarily long cells. For example, the part of your nerve cell that has the nucleus, where the DNA is located in your spinal cord. But it can extend a very long tube all the way, for example, to your foot. All of the building blocks for that nerve cell are made in your spinal cord, and all those building blocks have to be shipped to the very end of that nerve cell a meter away. There had to be some kind of transport system that was moving these building blocks inside the nerve cell, and I wanted to know how that transportation system worked. After we got this transport to work in this test tube, the hunt was on then to find the key molecule that was responsible for that movement. And we eventually found it, and it turned out to be something completely new that no one had ever discovered before. Watching these movements under the microscope, it was fascinating then to figure out, how does this motor actually work? How does something that's a millionth of an inch in size generate that motion? My lab at UCSF spent about 10 to 20 years trying to figure out the answer to that question. We know about kinesin now, we know a lot about how it moves, but there's still so many fundamental questions that we don't know about how all this motility is regulated, how all of these cargoes know how to go to the right places. There are always new questions that one wants to know the answers to.
Super cool. So pretty amazing, right? I mean, I think it's kind of amazing. Little tiny molecules moving stuff extremely long distances.
I was going to ask you a question, but it's not important. I'll ask you after class.
Yeah, okay. And then dyneins basically do something similar, but they go backwards, right? So kinesins move forward, dyneins move backwards. What else do you find within the cell? You're going to find Golgi output posts. And so Golgi outposts are going to be, doing a couple different things. They're going to be helping to basically, repackage contents that they receive, break things down. And you're going to find Golgi satellites. Those are going to be towards the dendritic ends. You're going to find a Golgi outpost that's going to be closer to the cell body. And then you're going to find the actual somatic Golgi that's going to be within the somite or the cell body of the neuron. What else do you find in a developing neuron? You're going to find lamellipodia as well as filopodia. Lamellipodia, that's going to be containing two major things, two major structures. It's going to have microtubules, which we just talked about, right? The scaffold, for example, on which dynein and kinesin can walk. And you're also going to find actin. Actin is going to be a much thinner filament than microtubules. Microtubules are much larger. So that's going to be lamellipodia. And you can think about it as like your hand, right? The lamellipodia, think of it as being like the webs of your fingers, right? And in the webs of your fingers, you're going to find both microtubules and actin in terms of the neuron. Then the fingers are going to be the filopodia. So filopodia comes from the derivative finger, right? So philo, finger. So there's going to be the finger-like projections at the very tip of the axonal growth cone. And those only contain actin filaments. And so here's a couple images over here that basically show what those different structures do, right? So you have the filopodia are trying to establish connection with different dendrites. And so, for example, this neuron over here, it's basically starting to form like 3-4 different dendrites until it latches on to the axons, until it latches onto a dendrite, and then it can basically prune back those additional axons so it can focus on the primary mode of communication, right? Through that one single axon. Again, microtubules and actin, those are going to be some of the major components of lamellipodia and filopodia. And the green, that's where you find staining of microtubules for these images. The red at the very tips, that's going to be just actin at the tips of the filopodia, for example. And here's an image showing the progression of growth of an axon in an embryo. And this took about 35 minutes for the axon to move 50 micrometers. So it doesn't seem like a long distance, but it's pretty significant over a course of 35 minutes. So this kind of stuff is happening all the time, right, in neurodevelopment. Here's an image from Cajal, right, in 1899. So he drew these pictures out. And basically, the growth cone is a way for the neuron to be able to decide what it's going to communicate with, right? And so you have the growth cone just trying to find a way to develop a synapse so you can allow for synaptic connection and communication. Now, in terms of growth cones and migration patterns, these are going to be really important to, for example, determine whether or not neurons are going to be ipsilateral, which means on the same side, versus contralateral, which means on the opposite side. And there's going to be different types of growth factors that are going to be affecting the way in which neurons can migrate, different migratory patterns. So here's an example using your eyes and the retinas and cranial nerve 2, right, the optic nerve. So there's going to be certain nerves within that tract that are going to remain on the ipsilateral side, and there's going to be certain nerves that are going to cross onto the contralateral side. And so there's going to be nerves that are going to be found within the nasal region and nerves that are found within the temporal region. Now, the way that the eyes work when they're receiving light, the nasal region of the eyes are going to be receiving image from the peripheries. So the images from the peripheries come into the nasal region, and then they get integrated into the brain, right? So we're going to talk about vision later on and how, vision is going to turn upside down, then it's going to turn right side up, right? Where you have different types of processing at different regions of the brain. The temporal sides are going to be receiving images from basically the medial visual field, right? And so they cross over. Now, which nerves remain ipsilateral and which nerves cross over? The nerves that are going to be found within the nasal region, those are the ones that are going to be able to migrate to the opposite side or the contralateral side. So temporal nerves are going to be ipsilateral. The nerves that are on the lateral sides, the temporal sides, are going to remain on the same side. The nerves that are going to be in the middle are going to be the ones that cross over to the contralateral side. Now, why is this something that might be relevant if you were to do like clinical practice on a patient that maybe was complaining of, for example, headaches, maybe changes in their vision, right? They might also have like hormonal changes, hormonal imbalances. They might have a pituitary tumor, and it's going to affect a specific region of your vision, right? It's only going to affect the vision that's received by the nasal portion of the retina, right? And that's going to result in lateral vision loss, right? Peripheral vision loss. Because if you follow the red lines, you see peripheral vision is going to be received by the medial portion or the nasal portion of the retina. And those are the fibers that cross over. And where they cross over is right next to the pituitary gland. So if you have a pituitary gland tumor, you're going to get some potential vision loss on the lateral side, right? Peripheral vision. Cool. So what are some of the things that affect the ability for cells to migrate, grow, form connections, right? You have a bunch of things coming into play, right? One of the major things is going to be calcium. Calcium is really important for all sorts of internal cellular communication, right? So calcium is critical in a lot of these processes. So calcium is going to be, for example, one of your second messengers. Now, you guys remember your second messengers from way back in anatomy and physiology. Right? You have like cyclic AMP, cyclic GMP. Calcium is going to be also another one of your second messengers. So in other words, a receptor has to be stimulated, and then calcium is going to be involved in the downstream cascade within the cell. You also have changes in direction of cells, right? The cells aren't going to always want to stick to their, whatever direction that they're moving in. Sometimes they're going to have to like change gears, right? Switch gears and move into a different direction. And an extension, of course. When you have axons growing, it's going to be a process of extension of that axon. And that's going to involve microtubules as well as actin, right? And microtubules especially are going to be undergoing depolymerization and repolymerization, right? What's a polymer? It's basically a string of proteins, right? That's what a polymer is. And so as the microtubule continually develops, it's basically like a giant swirl, right? You get all these little proteins that form a long coiled tube, basically. Cool. So here are your different filaments. You got your actin filaments, right? Those are going to be much smaller. You'll find them both in the lamellipodia as well as in the filopodia. Then you have microtubules that are only really found within the axon as well as the lamellipodia. Filopodia is going to be exclusively actin. So here's the difference between tyrosinated and acetylated microtubules. When it's tyrosinated, that's when it's dynamic and it's moving around. I got a couple cool videos here that I want to show you in a second. But then when it's stabilized, that's when it's going to be acetylated. So let's watch a couple videos on microtubules because it's really cool how microtubules are formed.
Meet the kinesin. Masterpieces of microengineering, kinesins are miniature motorized machines that carry cargo from one part of the cell to another, walking along self-assembling highways called microtubules. Known as the workhorses of the cell, kinesins have 2 feet, or globular heads, that literally walk 1 foot over another along the microtubule, pulling their cargo to its destination. Each foot possesses 2 special locations, called binding sites, that interact with other molecules. One site attaches to the microtubule, and the other binds with ATP, the energy molecule of the cell.
Okay, we already watched a video on kinesins and how they work, so let me skip that. Let me show you another video of microtubules. I like that video because it kind of showed, the way that the little tiny proteins start to assemble, right, as you get the polymerization of the microtubule. So this video is really cool because it shows as the microtubules kind of like retract and extend and retract again. So it's super cool. It's a dynamic process.
Microtubules continually grow from the centrosome added to a cell extract. Quite suddenly, however, some microtubules stop growing and then shrink back rapidly, a behavior called dynamic instability.
Okay, one more video. So again, you can see that the microtubules are basically extending, contracting, growing, moving about, changing directions. So super dynamic structure. And you find microtubules in all sorts of cells in the body. The microtubules are ubiquitous. It's not just neurons. They're going to be found in all sorts of cells, as well as when cells undergo mitosis, right? Microtubules are going to play a critical role in the process of mitosis. All right, so in terms of these growing filaments, right? You got your filopodia, you got your lamellipodia, and then you got the growth cone. This is all going to be important for the establishing of direction, right? Direction and extension of the cell. So here's an image that they're showing the growth of the growth cone over a span of several hours. And you can see that over that period of time, the direction started to shift, right? It started to grow into a completely separate direction. And that's going to be guided by both the filopodia as well as the lamellipodia. So what's going on in terms of all the signals? So remember, super important, you got calcium, right? So calcium is going to be one of the most important second messengers that come into play here. And as the growth cone continues to move forward, you're going to have a leading edge, right? The leading edge is going to be where the growth cone extends. And so you're going to have different cues that come into play that allow for the growth cone to move. So you have attractive cues that are going to be like chemoattractants, right? And then you also have chemorepellents that can also prevent the growth cone from moving in certain directions. So we'll talk about those chemoattractants and chemorepellents later on. You also have actin being constantly formed in these processes. Again, it's going to be requiring calcium for that to take place. And then you also have your microtubules being formed as well as broken down constantly. So repulsive cues and attracting cues are going to be really critical in the ability for the growth cone to move in different directions. So again, growth cones, they're going to require calcium, right? So it needs to have a rapid influx of calcium. And you can see in this image over here, it's showing one of these little filopodia and how it's changing in color here due to the influx of calcium. And so calcium is going to be integral in the ability for the growth cone to move in its direction. So here are your different major types of signals. So you're going to have chemoattractants, you're going to have chemorepellents, you're also going to have tropic molecules, and you're going to have trophic molecules. When we talk about tropism or trophic molecules, that's something that allows for extension and growth, right? It's going to be able to provide guidance and movement, the guidance of movement. Versus trophic molecules. Trophic is specifically going to be due to support, right? So you have to have, like, for example, nutrients and chemical support to allow for the neuron to remain alive. And these are all going to be working with different types of signals, like diffusible signals, receptors, and their respective ligands. So here is the difference between diffusible and non-diffusible signals. In terms of diffusible signals, which we've talked about even in like embryogenesis, these are signals that can basically move around the extracellular fluid without any issues. They can basically move freely. Neurotransmitters would be an example of this, right? Neurotransmitter gets released at the end of the presynaptic terminal, right? And it moves freely within the synapse until it binds onto the postsynaptic receptor. Non-diffusible signals, however, do not move freely within the extracellular space. And so non-diffusible signals, for example, well, netrin is actually a diffusible signal because it's moving through, but the receptors would be involved. The receptors that are involved would be considered non-diffusible. So diffusible signals, you have your netrins, which would be one of the ligands that get released, as well as the SLIT ligand, which binds to the roboreceptor. Netrin is going to bind specifically to the DCC receptor. So we're going to talk about those in a little bit and what they do. Other cues that are going to be coming into play, adhesive cues. So you want your neurons to be able to adhere to certain areas, right? So as the neurons grow, maybe as they scaffold, maybe as they form layers, you want the neurons and the axons to be able to adhere, right? Especially if you're like forming neuronal tracts. So you have adhesive cues, and then you have, again, your chemoattractant and chemorepellent signals. And then your trophic signals, which allow for this, the growing neuron to be supported with nutrients and things like that. So repulsive cues would basically steer the growth cone away, right? Chemoattractants are going to allow for the growth cone to move towards a certain direction. Chemorepulsion is going to push those growth cones away as well. And then you have your trophic support, which allows for the cell to have all the nutrients it needs as it continues to grow. Netrin, as you saw over here, netrin that binds to the DCC receptor. Netrin is going to be one of those types of signals that allows for attraction. So it's going to be a chemoattractant. And so if you were to have a lot of netrin being released, you're going to see lots of growth cones coming into play in those regions, right? So in this image of an embryo, you can see netrin is being secreted by a cell, and you're getting a lot of growth cones forming. Lots of neurites are going to be involved in that process. Here are some other signals that are important. These are, again, some of your diffusible signals that can just be released into the extracellular matrix. So SLIT as well as ROBO. These basically are different signals that allow for chemotropic signals, right? So tropism, right, meaning that you're going to be directing which ways those growth cones can move. SLIT, for example, allows for neurons to move across the midline. ROBO basically prevents those neurons that moved across the midline from moving all the way back again. And so WT, that means wild type, right? So here is normal, wild type, neuronal growth. So you had both functioning ROBO as well as functioning SLIT signals. If SLIT were to be disrupted, then you're not going to get crossing over the midline of different neurons, right? And then if ROBO gets disrupted, you're going to have crossing over the midline, but then those neurons are going to cross back over again. And so it basically disrupts the migratory pattern of these growth cones. So those are example of chemotropic cues, right? So tropism, the movement of, or the direction of movement for these growth cones. Netrin and SLIT, again, netrin is going to be binding to the roboreceptor, and then, SLIT binds to the roboreceptor, netrin binds to the DCC receptor. And what do these do? They basically affect the actin cytoskeleton. So they affect actin cytoskeleton, and remember, actin is going to be really important as part of your filopodia, the finger-like projections of the growth cone. Here are some other chemotropic signals. So you have Semaphorins, which are going to be chemorepulsive. These are going to be associated with plexin and neuropilin. Those are going to be the receptors, so we'll talk about those a little bit later. But sonic hedgehog, remember? Sonic hedgehog, super important for cell migration. If you have disruption of sonic hedgehog, you get conditions like holoprosencephaly. Right? So sonic hedgehog as well as WNTS, these both act as chemoattractants and chemorepellents. So they do both types of function. All right. So we talked about diffusible signals. Now we also have non-diffusible signals. And if it's a transmembrane protein, that's non-diffusible, right? That's basically going to be embedded within the phospholipid bilayer of the cell. And there's a couple of these that we're going to talk about here. We have plexins and neuropilins, and those are going to be associated with semaphorin. So semaphorin is going to bind to either plexins or neuropilins. And what do these do? Again, these also affect the actin cytoskeleton. So it's going to be important for the development of the growth cone. Again, calcium is going to be really important in this whole entire process. Calcium is one of the most important ions involved in neurogenesis and growth cone formation. So over here you can see how calcium, once you have the calcium influx, you get once you get the semaphorin being activated, right, then you get calcium influx, and that allows for guidance of the growth cone. Got 5 more minutes left. It's been a long enough day. Do you guys want to call it a day? Yeah, I can see you guys are like, I'm losing you. You have a headache? Okay, so do I. Right, so we will continue on here. So we're going to continue on slide 36, and we'll finish up all this on Tuesday. We'll run through both of the quizzes for this. And yeah, again, your first exam is going to be 1 week from today. So 50 questions. I've already started writing the exam. I haven't completed it yet. That's going to be 50 questions, multiple choice, Scantron. And as I mentioned, whatever I put in the quizzes questions that I produce, the ones that I actually make, not the ones that the publisher makes, but whatever I put in the quizzes, those are going to be the major topics that I focus on for the exam. Okay, it's not going to be the same types of questions. It's not going to be the same exact question, but it's going to be similar topics. Sound good? Rock and roll, guys. Have a great weekend. I will see you all on Tuesday.